Hello and welcome to MMI. In this series of tutorial videos, we'd like to show you the basics of the MMI CellCut and how to get good results for further research. The MMI CellCut is our laser microdissection system designed to cut tissues, large regions of interest or single cells, and even living cells for further processing. With its high-pulse UV laser for precise cutting and the patented CapLift technology for reliable tissue and cell isolation, it will help you with all your laser microdissection needs. In this video, we show you how to perform a serial section. This feature is very useful to cut the same structure or region from multiple sections of the same sample. First, we check that the current slide is the master section. Afterwards, we can start drawing our shapes the same way we showed you in an earlier video. The next step is to set 3 distinct landmarks on your slides. They should be recognizable on every other section of your serial section. The software will use these landmarks to identify the position on the master section. Once we're done drawing, we can switch to our next section. Now we have to tell the program that we are on a new section. Just click on the pen icon and then Add, and a new section will be created. Then we will have to find and mark our 3 landmarks again. Then we need to click on Transfer and the software will automatically draw our new shapes. The software will also automatically morph our shapes according to the landmarks we've selected. If they do not fit perfectly, we can move them a bit. Now you can repeat the process for other sections. We hope this video helped you with working with the MMI CellCut. Feel free to visit our other videos for more information about MMI instruments and handling.